Okay, hello. So here we are for chapter 11, actually nearing the end of the course load, nucleic acids, and it's subtitled Big Molecules with Big Roles. Actually, this is going to be pretty interesting. Here's where we're going to talk about the nucleic acids being interwoven amongst each other in terms of DNA and RNA fragments and the implications of some of that. So this is going to be a good chapter. I really like this chapter. All right, so let's just introduce this nicely. So here, you guys have heard of DNA. DNA is a molecule found in the nucleus of our cells, and that stores and directs info regarding cell growth and reproduction. A genome, then, is the total genetic info found in an organism. This includes DNA, which includes genes and non-coding DNA and/or RNA, such as that found, for instance, in viruses. So these are some of the vocabulary terms that I think are going to be very helpful in this chapter. Individual genes contain info to make a particular protein for the cell. Just so that you guys know, there's about 6,000 genes in the brewing or baking yeast, the Saccharomyces cerevisiae genome. That's not very much, but if you look at the fruit fly, it's more than double that. It's 15,500 genes. It further increases to 19,000 if we're talking about dogs. And then humans, where do we fit in? We're about 23,000, but guess what? The mouse genome has even more genes than us. And among all of them, the freshwater flea genome has about 31,000 genes. The Daphnia pulex. In fact, there's an outfit here at Indiana University in the state of Indiana that is actually investigating these kinds of species, these freshwater flea species. This is more of a crustacean, very tiny, and it is present in the lakes here in Indiana as well as other places. Anyway, the point is that it has a lot of genes, and so just as we have genes, we have almost about 23,000, and the number keeps on it's slowly inching upwards as more resolution of different experimental apparatus allow better resolution of the various genes, to be identified. Sometimes it's maybe a little bit less because maybe some researchers figure out, well, maybe this particular gene isn't necessarily, one of those that is encoding for DNA. There's a lot of non-coding DNA fragments within our genome. Although there is a temptation to call this quote unquote junk, it isn't junk. Actually, it's remnants of other parts of nucleic acids, including RNA fragments, but we'll get to that later. What is RNA? RNA is the middleman that links the gene from the DNA to the specific protein formed. And of course, the ribosome is going to be critical in producing those proteins, and we'll discuss that as well. DNA and RNA are huge molecules. Okay, RNA is smaller than DNA, just from a comparative perspective, but they are comprised of repeating building blocks called nucleotides. All right, and here you have, like a double helix here, representative of the DNA, and then you have the single helix representative here on the RNA. The nucleotides are comprised of nitrogenous bases, sugars, and phosphates, and that's where we're going to begin our discussion. With respect to nucleic acids, we have to talk about phosphate. Okay, most proteins comprise about 20 amino acids, sometimes a little bit more, and these nucleic acids are created from nucleotides, okay? RNA and DNA, this applies to both. You have your nitrogenous base, which is indicated here on the far left in this cartoon. You have a 5-carbon sugar, and I just tucked in the oxygen there just to show that it is a cyclic sugar structure, and really there's a carbon that comes off of that 5 position to give it that 5-carbon network there. You also have phosphate. So then if all of these condense together, right, if they are synthesized together in a condensation process, then you generate a larger molecule. Okay, nitrogenous base, we have pentose, and again I asterisk that area there in the 1, 2, 3, 4 position that has the 5th carbon extending from it as a methanol. Okay, and then of course you have the phosphate structure. What does the phosphate looks like? Just that, PO4, H, and it has a 2 negative charge, okay? And that's gonna become very important because nucleotides have the phosphate, whereas nucleosides will not. It'll become apparent later. All right, so we have nitrogenous base, we have the sugars, we have the phosphate. Let's talk about the different nitrogenous bases. There are 5 different ones, and these are either the purines, noted as either the adenine or the guanine, letter A or letter G. And this goes back to, having certain character within a given amino acid. So some of these might become apparent in some amino acid characteristics. And then you have the pyrimides, and those you have the cytosine, you have the thymine, and then you have the uracil. So these are the 2 different nitrogenous base types, okay? The purines or the pyrimides, and each of those then gives rise to either one of 5 nitrogenous bases that comprise DNA or RNA: adenine, guanine, cytosine, thymine, or uracil. Okay. Now, with respect to the pentose sugar, let's take a look at this. This is created from 2 possible pentoses, and these are going to become very familiar for you: ribose or deoxyribose. And this is where the whole deoxyribose nucleic acid gets its namesake, right? So you have either the ribose form or you have the deoxyribose form. Notice what that means then. If you have a deoxy, is that it lacks the hydroxide group, okay? And that's going to be at the number 2 position. By the way, as we start to number the carbons in nucleic acids, in these nucleotides or nucleosides, we start referring to each of these carbons with a prime. So we'll talk about 1' or 2' or 3'. That doesn't mean that it's primary, secondary, tertiary, and so on. That's just the nomenclature. That's just how we're keeping track of these carbon species, okay? And that's going to become even more useful in just a moment. I'll show you. But really, the look that we have to give this is having the primes attached to the numbers. 3 and 5 are actually going to become very important. You'll see why in a moment. But this is the basis then of having, this type of 5 carbon sugar, and you can see what I mean where you had that methanol group coming off that 4 prime carbon there, where I had asterisk above, and you have the methanol indicating. So that's where that is. So you have ribose or you have deoxyribose. The ribose is then present in RNA and the deoxyribose for DNA. In fact, that is what gives it its namesake. Condensation reactions then are going to form between a sugar, for instance, and the nitrogenous base. In a condensation reaction, you know that these are going to come together. It's going to form a larger molecule, and you're also going to think that water will be formed, right? Well, based on your sugar chemistry knowledge, where do you think the condensation will occur? In other words, which carbon on the sugar molecule might be affected? It's actually going to be affected at that 1' position, okay? And it's also denoted to be that anomeric carbon. If you remember, within the cyclic pentose, that was done through a hemiacetal, but you can go back there later, but we're not going to dig that up too much right now. What we're going to be focusing on is just going forward with this. So here we have the nucleoside. You can see how the bond forms between that sugar group and the nitrogenous base. That linkage then is that glycidic bond, okay? This nucleoside, by the way, has the character of adenosine, okay? And again, you still had water being produced, and I have indicated there that that bond is in fact the glycosidic bond. Again, know that the nucleosides do not have a phosphate. If it did, then you would call it a nucleotide, okay? Here would be a blow up of that reaction where you have adenine, okay, interacting with ribose. That is that is the reaction here if we're considering condensation. The H from the adenine and the OH group from the ribose are going to be split away from that, and thus water form. So there's your cartoon there showing a little bit more the actual structure, skeletal structure of what's going on. However, in this particular case with the sugars, some of the hydrogens are shown as well. So I think that kind of helps understand where and what is going on. With respect to the condensation, then, if we're looking at the phosphate then, and then the nucleoside, what does that look like? Well, again, this is going to form yet a new nucleic acid molecule. This is going to be considered then a nucleotide because of the presence of the phosphate. So the phosphate's going to link up with the sugar, okay? And where do you think that's going to occur? That's going to occur at that number 4 position, right, where you had that methanol that was attached. In this particular case, here you have the phosphate attached then to the sugar that had already been attached to the nitrogenous base in the nucleoside environment. Now, this new bond is called a phosphoester, all right? And water is still produced because it is a condensation, but instead of a glycosidic bond that exists within that sugar or between the sugar and the nitrogenous base, which still exists, the new bond that links the phosphate to the sugar entity is going to be called the phosphoester. And where do we get that? That's coming from the ROOR functional group. So you're getting at least one of those R's from the sugar, okay? And if we have to blow this up using the actual chemical shorthand here, we're going to use these skeletal structures with some of the hydrogens drawn in with the hydroxyls. Here's what we're looking at. You have this phosphate, and it's going to be attached to the ribose, okay, at that number 5 position, all right? And in doing so, then the hydrogen will split off as well as the OH group from the phosphate entity. That will form the water entity. Meanwhile, that CO, right, remains, of course, with respect to that sugar, and then the phosphate links up. The ROR kind of bonding, that's the phosphoester type of bond, okay? So that's just the nomenclature of what that's going to be. So just know that if you have a POR group, which could stand for the rest of the molecule, we're looking at not only that methyl group there, but the rest of the sugar entity as well as its attachment, obviously then to that nitrogenous base. You can call all that the R group if you so wish. But the fact of the matter is it is a phosphoester type of bond. Instead of an ROR bond, you have a phosphate OR bond. So kind of interesting. Okay, so let's talk about the condensation then of a nucleoside. So here we have, again, a sugar molecule, right? 5-carbon pentose, right? And then you have a nitrogenous base, okay? One of these, this is the pyridamine, and then you have the ribose, and they're going to come together in a condensation reaction. Where does that reaction occur? It's going to occur at that anomeric carbon of the sugar. So the OH is going to pop off there. The hydrogen is going to be given up from the 1 nitrogen, number 1 position nitrogen on that pyrimidine molecule, okay? And it's going to link up, and then you'll have your nitrogenous base connected to your sugar entity, okay? So there's your nucleoside formation. And again, the glycosidic bond is there. What about the nucleotide? Again, it's going to happen in the same place. You have the phosphate coming together with that methanol. That was part of that whole 5-carbon pentose structure there. And that would be the deoxyribose, right? And so what you're getting then is that phosphoester character whenever they link up. Again, nothing changed with that glycosidic bond. Water was still formed. One of the H's came from that methanol group from the sugar, and while the OH group was given up from that phosphate, okay? So here we have the 5 types of nucleotides. There are only 5 that we're going to consider here, and that are ubiquitous actually in most living forms. So we have the adenosine monophosphate. It's indicated as AMP, okay, if we were to abbreviate that, and that would be with respect to RNA, whereas the deoxyadenosine monophosphate will correspond to DNA, okay? Guanosine monophosphate, GMP, and deoxyguanosine monophosphate, DGMP. Cytidine monophosphate, CMP, with deoxycytidine monophosphate, DCMP. You have uridine monophosphate, UMP, okay, which is only going to be present for RNA. And then you have deoxydimidine monophosphate, DTMP, only available there for DNA. All right, so the black is written as the RNA molecules, and the red is indicating DNA molecules. So notice that although you have 5 different types of nucleotides, there are still 4 per RNA, 4 per DNA molecule. Okay, so the uridine monophosphate, the UMP, is not going to be present with respect to the DNA. Likewise, the deoxydimidine monophosphate, the DTMP, is not going to be present, meaning found in the RNA. unless of course something was artificially spliced in there and then it, you know, things like that were forced. So we're not talking about engineering from that point of view. We're just talking about what is commonplace, okay? Again, the deoxyribose indicates that you have DNA. What is that from? Well, that's an H substituting the OH, okay? And the H's and the OH's, they're shown here, you know, where that hydroxyl group would be, or that hydrogen group would be at the 2' position within that ribose molecule, okay, the sugar molecule. And so you have the ribonucleic acid, again, RNA, deoxynucleic acid, DNA. Okay, so I think that that's pretty clear. In this particular case, we're looking at table 11.1. This is given in your book, nucleoside versus nucleotide names in DNA and RNA. And it's everything that I just outlined for you there in those schematics, all the different bases, the nucleosides and or the nucleotides, okay? And the ones that obviously have a D, E in front of the, well, it's the deoxy actually, the whole bit refers to DNA, and those that don't, that just have the ribo, you know, nucleic acid formation, the RNA, won't have any of the deoxy anything. In fact, they won't even have a ribo present, and it just says, you know, it'll be uridine monophosphate or, you know, cytidine monophosphate, guanosine monophosphate, or adenosine monophosphate. That's just what it is, okay? Again, the nucleotides indicate that you just, that you have the whole combination of nucleic acids with 3 components, right? The nitrogenous base, the pentose, as well as the phosphate. Nucleoside will lack that phosphate. All right, so here are some different components, you know, from a cartoon perspective. If you looked at DNA on the top and the RNA on bottom, starting from the upper left-hand part of this cartoon and working our way down, if that were to break down, you know, undergoing hydrolysis, this could be part of digestion, for instance, and they would form nucleotides upon initial hydrolysis. Further hydrolysis would further break these into their individual component species. So we're going from nucleotides to, for instance, the bases, the sugars, and the phosphates, okay? If we were to do this reaction, what would that look like? So we have the dinucleotide, okay? And this is, again, with phosphate formation via condensation. So we have this nitrogenous base in the upper left there, the green, right, connected to the sugar, right, the ribose. And in this particular case, it's going to be the deoxyribose, right? But it doesn't have any kind of indication about where they might link up until we start seeing, well, if there's another molecule in place here, then maybe there can start to be some similarities, and we find that. So the positions that we're going to be looking at, let's ignore the nitrogenous base for a while and just only focus on the sugar. In the upper left-hand image there, you have, meaning the yellow sugar, you have the number 3' position indicated above the hydroxyl group. And then you have the number 5 position of that methyl group that's attached to the phosphate in that phosphoester bond. Those 3 and number 5 numbers are actually going to become super critical, and that's going to dictate where the reactions are going to be whenever these individual nucleotides are combined, whenever they are condensed. And so in this particular case, you have deoxycytidine monophosphate, which you knew was a DNA-based nucleotide because there lacks the hydroxyl group at the 2' position in that ribose molecule. And then you also have deoxyadenosine monophosphate, the DAMP, DAMP, right? And that also is a DNA-based nucleotide just because of the, again, the lack of the hydroxyl group there. If those two are going to combine, they're going to form a dinucleotide, okay? And where would that be? So you have the upper molecule, the deoxycytidine, combining with the deoxyadenosine. Again, ignore where the bases are, and let's just focus on the sugar entities and where those are. Notice that the hydroxyl group from the number 3 position coming from deoxycytidine, okay, the topmost, is going to lose its hydrogen, okay, and it's going to be able to, in fact, you know, you could even say that it lacks, that's going to lose both the hydroxyl group there, all right, and it's going to be able to interact with that phosphate group, okay, from the deoxyadenosine. In that particular case, now a new bond is formed, all right, and you have then, in essence, another phosphoester. You would call this grouping of the phosphate group that's sharing 2 different phosphoesters, right? One with its existing nucleotide structure, the adenosine, where there already existed a phosphoester, right? Just like how there already was a phosphoester in the deoxycytidine. But here, the sugar entity, that pentose from the deoxycytidine, formed a new phosphoester with the other nucleotide, the deoxyadenosine, at that same phosphate, species. Not the exact same position, obviously, because one of the oxygens is already taken up by that nucleotide, deoxyadenosine. So the other one is going to be the deoxycytidine, is going to link up with that phosphate species. And so doing, you have a phosphodiester. That number 5' never changed throughout this whole process from that deoxycytidine nucleotide, okay? We would consider that to be the quote-unquote free 5' end. Meanwhile, in the ribose, in that 5-carbon sugar molecule, right, in the deoxyadenosine, that is also free because the hydroxyl group still exists, okay? That's also called then the free 3' end. This is just the vernacular and this is just the vocabulary that we just have to be comfortable with if we're talking about growth, right, of nucleotide formation, because in so doing, this is where you can get long chains then of molecules to be able to be developed, provided there is mechanisms then in place to allow for that condensation, okay? So the phosphate group of only one of these nucleotides participated in the condensation reaction. Notice that although the hydroxyl group popped off, right, and here it's looking like, again, experiments may show that the phosphate loses that oxygen, but it can also be possible that the hydroxyl group is lost from that ribose in order for the formation of water to occur, okay? It might be just slightly statistically more favorable for that oxygen to pop off, okay? Studies have already shown these things. You can label them, you can follow them, and then you can figure out where those interactions are. That's just, one of the ways. But I think it helps to know that regardless of where that oxygen is coming from, you're going to have one of the oxygens coming from somewhere, whether it's from the phosphate or the hydroxyl group on that number 3 position of the sugar. It does not matter so much for our purposes here. Knowing that you're going to have an H and an O coming from that region, it's going to be, combining with a free proton that already was in existence or in that environment. Where is that coming from? Well, that could be a proton that was provided by maybe a cofactor reaction where there was some redox going on. That could have been provided, for instance, in an acidic environment, for instance, like in the stomach, where there is an overabundance of protons because of that acidic nature. So the generation and the presence of that proton is coming from another source. So it could just be its environment, could be produced from a redox reaction. We're not really concerned about where that's arising from. What we are concerned about is knowing that water is formed. Nothing is strange about this. You still have one hydrogen coming from the environment in which these individual nucleotides are existing whenever they come together to form this dinucleotide, and then the other hydrogen and oxygen that are coming from the actual nucleotides themselves, okay? Now, if we were going to express this dinucleotide then as a sequence and name it using 1-letter base codes, which is what scientists do, this is what is found in various research papers, you would first identify which end is the free end of the 5' and then the 3', okay? And again, what is this doing? You don't worry about the nitrogenous base or the phosphate per se, you're just focusing on the pentose ring. So just focus on what is yellow, and in those yellow regions then are what's going to dictate where the free 5' and the free 3' might be, okay? So the 5' free is going to be found at the deoxycytidine. The 3' free is going to be found within the deoxyadenosine, okay? And so then you write the backbone and you use the nucleotide names. So you have the free 5' end, right? You have obviously the phosphate, you have the sugar, you have the phosphate, because that's what it's linked, phosphate, all that stuff. And then you have your free 3' end. That's the backbone. The bases come later. Those are the offshoots, okay? And what you do is you just label these then using the nucleotide name. You have the cytidine and then you have the adenosine, okay? And then use the one-letter codes. Knowing that these are the cytidine and the adenosine, you would then write this then as the 5' of the cytidine, which is denoted as the letter C for that cytidine molecule. And then you have the adenosine, right, as just the one letter A representing that molecule. It's 3N discrete. That's how you would write it. 5' cytidine, adenosine, 3', or 5' CA 3'. Okay. Here would be another example. The nucleic acid backbone, given this, we're going to consider this to be then the primary structure. You have the phosphate and sugar and phosphate and sugar and phosphate and sugar and phosphate and sugar. Okay, you have 4 sets in this backbone. We're just looking at that. The phosphate on the far left is going to be the free 5' end, and the sugar is going to be the free 3' end on the far right. Depending on what those nitrogenous bases are, that's going to dictate what the sequence is going to be then, right? You either have G, you have T, you have C, and then you have A. All of those are then grouped together, but you don't worry about writing the phosphate and the sugar molecules because those are the same, okay? Those are just repeated. So you just say, well, the 5' is based on the phosphate group with the sugar, but it's all about that nitrogenous base at the end. What is it? It corresponds to G, guanine, okay? Then you have T, thymidine, cytidine, and then again you have adenosine, G, T, C, A. And the right side is going to have that 3', and the left side is going to have that 5'. You're never going to see, well, at least in your initial coursework and stuff, and I don't, I haven't seen this at all. Usually that 5' and 3' switch, it's going to maintain that integrity. 5 on the left, 3 on the right. And it has analogies to amino acids where you have the amino group generally on the left and the carboxylate group on the right. I should say the protonated amine, right, is on the left, and then the carboxylate is on the right, where the protonated amine is positive and the carboxylate is negative. So these are some of the analogies that you can draw from if we're going to consider this kind of nucleic acid backbone structure with respect to amino acid sequence, those connections whenever peptides are formed, polypeptides, and ultimately if you get larger than than 50 polypeptide or 50 individual amino acids, then you start talking about proteins. Okay. So again, this is the example, and if you were to draw that skeletal structure, right, indicating, though in this particular case you are putting in some hydrogens, so it's not a true skeletal structure, but you are denoting where all of the main functional character groups are, you would see that the phosphates and stuff would link up. In this particular example, you would only have 2, right? You have phosphate, sugar, and you have a phosphate and you have a sugar. You have 2 of the same nitrogenous bases, so this is going to be the same, okay? It's the same, they're both adenosine, all right? And again, it's based on that. We know that, we're just looking at that nitrogenous base. Basically, those cyclic nitrogen structures, okay? You have your free 5' end and you have your free 3' end, okay? You use the 1-letter base codes. What kind of nucleotides are we talking about? We're talking about the adenosine, okay? It's 5', AA, 3', or simply, because everybody in the field, you might say, well, why do we even have to use the 5' or 3' if everybody knows what that is? 3' on the right, 5' on the left. Well, then you don't. You just write AA. Here's nucleic acid backbone sequence and a naming example where we have 1, 2, 3, 4, and don't drive yourself nuts, just count either the nitrogenous bases, which there's 4 different ones, or the sugars, right? There's 1, 2, 3, 4 of those pentagram shape, and then, or you have the phosphate groups, 1, 2, 3, 4, the tetrahedral shape, right? So you have 4, okay, of these nucleotides, right? And so you're looking at that methyl group that's attached to that phosphate, right? Forms a phosphoester. That's the free end up top. Guess what? Very bottom end where the hydroxyl group is on the 3', that's the free end. So then it's just a matter of knowing what those nitrogenous bases are referring to. So top one is guanosine, the other one's cytidine, uridine, and guanosine again, okay? And so again, if you were to write this, it'd be GCUG. You could code that as 5' and 3' on either end, or you could just use it as GCUG, okay? So that shouldn't be too challenging. If we're going to talk about the secondary DNA structure, this is where things can start to get a little bit interesting, because we talked about that any kind of helical structure, especially in DNA, is going to be held together with hydrogen bonds. That's the type of intermolecular force, right? So let's take a look at that. There's only going to be certain combinations that are going to be allowed. So two nucleic acids have backbones arranged in anti-parallel helices, okay? One's going up and the other one's going down. So they're not both all going up in the same direction. That's why the arrows here at the top and bottom of this cartoon in this left image here are showing you that one wraps up 10 points to the right and the other one is wrapping down and points to the left. That is what we refer to as anti-parallel, okay? They're just opposite in direction. The sugar phosphate is going to give you the ladder rails. So all of that ribbon, all that nice flat pink ribbon that you see, it's basically the sugar phosphate groups. We just smoothed that out and just said, well, the sugar and the phosphate groups aren't going to change. That's the ladder rails. The stuff that is going to change and that is of primary interest here are the so-called ladder rungs, where you have the hydrogen bonding that is being able to pull together these different nitrogenous bases, forming a pair, because there can only be certain combinations of these bases that can work, and that the hydrogen bonding pulls that together, allows it to have a strong enough stability that it appears as if it's a uniform kind of ladder rung. If you're on a ladder, you know what I'm talking about. It's a pretty much a solid piece. Is it truly that in nature? No, but it acts like that by virtue of those hydrogen bonds, okay? Remember, the hydrogen bonding was like the third strongest intermolecular force, next to the London forces and the dipole forces. And then you had the hydrogen bonding. The next strongest was the ion dipole type of intermolecular force. Here we're not involving that because there aren't any ions that we are talking about within the structure, okay? So if there can be a certain set of pairs that can come together by virtue of hydrogen bonding and be established as like a quote unquote rung on a ladder, then there's going to be rules that we're going to end up following. So there's going to be a pyrimidine complementary base pair rule. That rule is that you're going to have adenine and thymine being one complementary base pair. The other one is going to be the guanine cytosine. Okay, so you're not going to have pyrimidine and pyrimidine combinations or purine and purine combinations. You have to have both representations of nitrogenous bases from each, the purine and the pyrimidine coming together to forming that complementary base pair. Adenine and thymine is one example, and guanine and cytosine is the other example, okay? Again, these helices are connected via hydrogen bonds, okay, between base pairs, which consist of purine and a pyrimidine, nitrogenous base. If we were to talk about the DNA double helix, then you could take another example of this, and you could either write it with respect to the skeletal structure, or you could show this in a cartoon with ribbons, and then you could just highlight that nucleotide base pair, that nitrogenous base pair, because really that's the difference then amongst these different, N groups, right? These nitrogenous bases, the thymine, the adenine, the cytosine, the guanine, okay? It's also indicated here where they use phosphate as pretty much a darker part of that ribbon within that helix, and then they use the deoxyribose sugar as the broader, semi-transparent but still purplish segment within that ribbon. And then of course you have the hydrogen bonds, okay? So there's a significant amount of hydrogen bonding going on. So whether you have it, arising from the hydrogen that's getting attracted to the oxygen, for instance, on a carbonyl, or you have the hydrogen being attracted to an existing nitrogen group on that nitrogenous base, as shown in the upper part of that image, it doesn't really matter, okay? So again, you have adenine, which is a type of purine, thymine, which is a pyrimidine, they are attracted. You have the adenine, thymine complementary pair. Likewise, you have the guanine, which is a purine, and then you also have cytosine, which is a pyrimidine, and they form the other complementary pair. All right, so let's figure out in this example. You have adenine and you have thymine, okay, and you have guanine and you have cytosine. All right, if you have the sequence, let's just say you have the sequence, and the sequence can arise through experimental methodologies that allow sequencing. We're not going to get into that, but if you want to know more about that, you can definitely reach out and ask, and I could, share some knowledge on this. But this is a key part of trying to sequence anything, as long as you have some kind of molecular information about that thing that you're trying to sequence. Okay, usually it's going to involve trying to scale up or reproduce whatever small sample that you have, and then you're just going to do several experiments, and you're just going to follow basically what looks like to be a race amongst different molecules to get from one side to another side, like a positive versus a negative end. You might have different snippets of different lengths, and they're all going to race across from the positive to the negative. And so that's just how some of these experiments can work. But anyway, so let's just say in one of those snippets you had this sequence 5', and of course bookended on the other side by the 3', you have A, C, G, A, T, C, T. And you're asking yourself, what is the complementary strand? Well, you know that adenine is a complementary pair with thymine. It is not with another purine, i.e. guanine, or the other pyrimidine, cytosine, okay? It doesn't work. So it's pretty easy. A goes to T and G goes to C. Pretty cool code, pretty simple. That shouldn't be too hard. So it's T, G, C, T, A, G, A, okay? That's the complementary strand. And that would give rise with respect to the double-stranded helix that represents DNA. All right. Finally, tertiary structure, right? Now here's where we start talking about chromosomes. If you have your DNA base pairs all matched up and you have these nice hydrogen bonds, in this particular case, these hydrogen bonds are indicated by these very solid ladder rung-like structures, right? These rods in this cartoon. Are they truly that strong and directional? Not really, but for intensive purposes, trying to show what's going on here, they are, okay? And they're wound about each other, all right? But what is giving rise to having all this DNA be wrapped about something? Well, you need to have something like some kind of a base material that serves as a scaffold, if you will. And so the histones do that, okay? And in doing so, then the DNA can wrap about that. Now, it will involve twisting, and supercoiling is going to be found here, where there's a super amount of twisting of the double helix, okay? Maximum amount. I mean, again, the focus here is really on the hydrogen bonding part, because that's what's really going to be driving all of this coiling kind of activity. I mean, that's what gives rise to the DNA. And then you might have some other types of intermolecular forces that are really going to try to further drive in that kind of super tight coil. Some of it could be due to hydrophobic interactions, all right? Because getting some kind of nicely arranged superstructure like this, the tertiary DNA structure, is going to require more than one type of intermolecular force. So just knowing that there's 3 billion base pairs that comprise one human cell, that's just crazy, right? It's about 6 feet in length if it's stretched out. It's massive. And that DNA in the human cell comprises 46 pieces, right? 23 pairs from each, mother and father, and they're super coiled about these proteins. And those proteins, of course, are proteinaceous fiber. Because it is protein, you're going to have the presence of potential London forces, like those hydrophobic forces, and you might also have some ionic based, like ion dipole type of intermolecular forces, depending on the nature of the histones, okay? But all that is going to give rise then to this kind of superstructure, this tertiary structure. Again, it has a combination of multiple different intermolecular force environments to be able to create such a structure like that. These DNA and histone supercoils pack into what we call these chromosomes. Now, an interesting case of this, for instance, and I'm getting this data from the National Library of Medicine. If we were to look at the different chromosomes, right? The 1, 2, 3, 4, 5, you just keep counting these. One is coming from the father, one is coming from the mother. These are definitely paired up. And then you have your germline, right? Your sex chromosomes, the X, and then of course the Y, and that of course dictates the gender. What would that look like if you were to try to map that out in terms of actual proteins and stuff? Well, you can perform experiments. You can understand that, with respect to that first chromosome, right? That chromosome number 1, we'll call it. That has it appears like it's the largest, and in fact it is. It has the most genes. Those genes, are wrapped into 2 different parts, a short arm part and a long arm part, okay? The short arm part is called the P part, and the long arm part is called the Q part. Interestingly enough, and so what we did here is I have the number 1 dotted here with that structure, and then what we did is we basically flipped that to its to the horizontal orientation. All right, so we took that vertical position of chromosome 1, switched it to the horizontal. You see that you have short arm, and then you have long arm, and then you have certain codes that are discussing each of these regions within the short and long arm respectively, and those are gene bands. And those gene bands can give rise to certain information about whether, for instance, there might be a risk for carcinoma in a given subset of your proteins, right? Usually this is going to be with respect to somatic stuff, things that have developed over time, and you assess it. You go, well, let me take a look at this. Let me see if there's markers for any kind of concerns that there might be in your body with respect to a certain organ. But you can definitely have certain analyses done. Of course, nothing is concrete, and it's all going to be based on probability, right? But generally, it's a decent science, and when I say decent, is that the accuracy can still be so high that you can have pretty good confidence in what you're looking at, and in which genes that there could be problems that may or may not be present. So there's, just so that for example, chromosome 1, which is the largest of the 22 pairs of identical chromosomes, plus then the 2 germline, the sex chromosomes. Chromosome 1 has comprises about 8% of the total cellular DNA, which is about 249 million base pairs. And it has between 2,000 and 2,100 genes that code for proteins. And again, these are just some of the bands that are being highlighted here in this horizontal segment or in the vertical segment. It doesn't matter, but all within that band, within that width of those colors there, whether they're going from light gray to darker gray or even to black or whatever, that means that there's other genes that are present within that band that may have similar properties. Okay, so here's an example of a gene producing protein, AGTR1. This is a gene that resides in chromosome number 3. Okay, it produces angiotensin 2 receptor type 1, an AT1 receptor. Why is this even important? This is involved in the regulation of blood pressure. And where would it be found? It would be found here where the yellow arrow is, this Q24. Okay, so that would be the band region, and that would correspond to that type of gene. Again, that gene is going to be called AGTR1. And here's a little bit of description about it that I wanted to share with you. And you can find more about this in the U.S. National Library of Medicine. And here, we had searched just for AGTR1, but there's other ones that you can look at too. But here would be a description that would discuss, well, why is this even important? So through a series of steps, the renin-angiotensin system produces a molecule called angiotensin 2, and that attaches, i.e. binds, to the AT1 receptors, stimulating chemical signaling. What happens then? Well, that signaling causes blood vessels to narrow, meaning they constrict, which results in increased blood pressure. So binding of angiotensin 2 to the AT1 receptor also stimulates production of the hormone aldosterone, which triggers the absorption of water and salt by the kidneys. So the increased amount of fluid in the body also increases necessarily then blood pressure. So that binding, which triggers, right, the production of this hormone, which in turn triggers the absorption of water and salt, would then lead to an overall constricted case, a very frustrated case where, blood vessels are narrowing along with a concomitant increase in water and salt absorption by the kidneys. So you would think that with the vessels constricting and with the higher absorption of water and salt within the kidneys, that might increase blood pressure. So, and in fact it does. So what can this do? What can this gene analysis do? Well, it can seize it. It can detect that there's any kind of abnormalities in that gene sequence, which would give rise to this. So that's one example. Now, if we're going to compare proteins and nucleic acids, we could do that, and we could start looking at, comparisons with respect to the primary structure. So remember, whenever we talked about proteins, it was sequence of amino acids. Here for nucleic acids, it's the nucleotides that we're interested in. And if you're talking about secondary structure, again, hydrogen bonding between backbone atoms. Well, guess what? There's also hydrogen bonding between the bases in the nucleic acid strands. So that is actually still the same. With respect to the tertiary structure, you still have folding of the backbone, but guess what? You're getting this massive, superstructure called chromosomes if we're talking about the nucleic acids. If you want further differentiation or similarity, actually, between the proteins and the nucleic acids, and they should be quite similar because you can't get the proteins without the nucleic acids, then you're going to look and see, well, the repeating unit of the protein is the amino acid. For the nucleic acids, whether it's RNA or DNA, it's the nucleotides, okay? The backbone repeat is the nitrogen and carboxylic groups, right? And here, instead, in the nucleic acid, you have sugar phosphate. One letter abbreviation, well, it works for amino acids. It can also work for the base with respect to nucleic acids, amino or N terminus, 5' end, free end is whether it's the carboxy or C terminus, indicated by the free end. So those are the analogies that we can draw there. Now, the interesting part about the DNA and RNA is this. Very few human genes, probably about 1%, some say up to 5%, but I don't know about that, they actually code for proteins. And this goes back into some of the non-coding kind of material that is present within our cells. Most are then called these introns. And these are promoters, enhancers, silencers, insulators that affect protein binding sites. And importantly, they also have other functions that are not fully understood. And again, where am I getting this information? I have to rely again on the National Library of Medicine just because it's a great resource. I mean, they really compiled a lot of different references all into one area. And this has been, it's a result of a large effort, obviously, going into this. But, these things are still being investigated. If we're looking at it from a research perspective or an interesting avenue of research, this would be one of them, right? Knowing that, you only have maybe 1%, but, let's just say maybe 5% of all the genes actually code for proteins. I mean, that is unbelievable. So all the proteins in our body that give rise to our tissue, to, right, all of our sensors, all of those different protein functions, enzymes, right, your skin, cell membranes, right, transporters, hormone, I mean, all of this stuff. Only 5% of our genes are coding for all of the proteins in our body. That is absolutely amazing. What is the other 95% of our genes doing then? That's really interesting, and this is a good area of research that anybody could get into. So in contrast, virtually more than 95% of bacterial genes code for proteins. That is incredible. So you're saying only 5% of our human genes can actually, code for proteins, whereas 95% of bacterial genes code for proteins. Well, no wonder why we're using bacteria as our little bioreactors to be able to generate really interesting things, whether it's something as simple as a sugar or maybe something more useful, perhaps, like an antibiotic. This is really interesting stuff. And it's really interesting because bacteria have far less genes. They only have about 2,000 total. So it's a hugely efficient system. That is absolutely amazing. Okay, so between RNA and DNA, we already talked about the difference between that uracil versus thymine base, that nitrogenous base. The sugar is going to be ribose. Guess what? For DNA, it's easy. It's deoxyribose. The stranding is a single strand of nucleic acid, whereas you typically have that double strand. For the DNA and the size, well, RNA is much smaller. So again, I'm going to remind you that the RNA is the middleman in the protein generation process by DNA. All right, we have three types of RNA. You have that are very familiar to us, right? You have the messenger mRNA, you have the ribosomal RNA or little r RNA, and then you have the transfer RNA, the tRNA. Sometimes I even like to think about that RNA transfer one as like a tag one. I'll show you why in a moment. But what's really interesting in this, before we get into this too deep, I want to have just a short discussion on this because this is absolutely amazing. These ribosomal and transfer ones have some really interesting backgrounds. So, and here, if you go to the DNA Learning Center or even the U.S. National Library of Medicine, you're going to find some really good references here. If we can look at mitochondria, all right, and here is like an SEM image or a microscopy image. You have mitochondrion indicated there. It's like that purplish kind of organelle, right? It's part of the eukaryotic cell. It resides outside the nucleus, but it's still within the cell, right? Look at this eukaryotic cell that has the nucleus, that has the mitochondrion, and then look at the upper right image of the prokaryotic cell. This would be an example of bacteria, for instance, E. coli. Look at the shape of the mitochondrion in that it shares then with the prokaryotic cell. I'm not saying that mitochondrion is in the prokaryotic cell. I'm saying that its shape of the mitochondrion that exists in the eukaryotic cell is really similar to an entire prokaryotic cell of this bacterial species. So geez, because of that size and shape similarity, Ivan Wallin in 1920 suggested bacteria as the origin for eukaryotic organelles, i.e. the mitochondria. That is absolutely incredible. So somehow the mitochondria, maybe one day, way back when, or maybe is in fact now, you never know, depending on your perspective, where you want to go with this, it is bacteria based. And what's especially interesting is that mitochondria are also known to have DNA. They call it the mtDNA. So mitochondria actually have their own specific DNA that is unique to that mitochondrial unit, that organelle, relative to the rest of the cell. So this, in my opinion, and it would seem based on this research, would further support the fact that, well, you know what, if there's a specific organelle that has its own DNA, yeah, it might have some really interesting origins in the history of life. And it might have some bacterial origins, in fact. So just to let you know, 37 genes, or about 16,500 DNA base pairs, right, are comprising the mitochondria. And these 13 genes, though, of them, right, contribute to oxidative phosphorylation, i.e. cellular respiration, because we know this is where the action is going. And it does so, of course, via enzymes, for instance, like an ATP synthase. So you need proteins in order to get this oxidative phosphorylation. But here's the thing, that's how cells are breathing. 13 genes of those 37 are contributing to that, okay? And this, eukaryotic cell, what's its size? It's quite a bit larger. It's about 100 times larger, right? And of course, that glucose and oxygen, generates energy and carbon dioxide of water, in the glycolysis, right, of glucose. And so you have to ask yourself, well, what about those remaining 24 genes? Well, they form the ribosomal RNA and the transfer RNA. And that is amazing. So the mitochondria is a powerhouse. We know that, but it may share common origins with the humble prokaryotic cell. Incredible. What do we mean by this? Let's take a further look, and we're going to go back to insulin again. We already had discussed that before, right? And we knew that was a protein, and we know that's involved in the regulation of glucose, right? Well, here I just have some cartoons in the topmost part that talk about the insulin dimer versus a hexamer, meaning that there's 6 of these insulin molecules versus 2. That's on the left, and then you have a different kind of blown up region, this ball and stick model, really. In the upper right hand image, it kind of shows this so-called A chain, charged surfaces, hydrophobic core, as well as a disulfide linkage, given rise by virtue of that yellow area. Again, to let you know, and we've already talked about him, but Fred Sanger, he won 2 Nobel Prizes in the 50s. He demonstrated that insulin's 51 proteins could be sequenced, and his experimental methods continue to be used today. He was so focused on using and exploring DNA while everybody else was obsessed with RNA. Why were people obsessed with RNA? Well, RNA is much smaller. They thought it would be much easier to investigate. In fact, it proved out that DNA, it's right. I mean, you have a double helix compared to the RNA single helix, and DNA molecules are generally larger than the RNA, virtually in every case, okay? So why would you even what would even make you want to study something that was even more complicated? It was the fact that DNA required RNA for templating purposes whenever there was going to be mRNA kind of development. So I know I'm talking about all these different acronyms and everything else, but the fact that RNA didn't require a template kind of led Sanger to think, well, hey, maybe we should be looking at DNA. Maybe I could try to figure out what the sequencing can do and how that relates to the actual proteins that are formed. And so that kind of thought, and he followed others that were also working in that space. So he was just another scientist that was showing good form, trying to be open to different ideas. Didn't have to follow everybody's idea, because a lot of the researchers during this time were exploring RNA, because obviously it was a little bit simpler, and in theory it could be better understood. But to be able to actually use RNA to his benefit to further explore DNA was something that made him famous in this field, to the point where his experimental methods are still being used today. And finally, in 1999, the Sanger Center finished sequencing chromosome 22. That was the smallest and the first one ever sequenced. And so it's been a while. So you have all the chromosomes that have been sequenced. It's been known for a while, but all this goes back to, well, how do you probe this? How do you sequence all of these different proteins and such? And so, like in this cartoon here, I'm showing you the amino acid sequence: leucine, valine, glutamine, alanine. You're seeing all those spheres, and they are representing specific amino acids, right? And then combination of those amino acids are going to give you that peptide sequence that you're interested in. If you have 51 of them here, in a certain arrangement, then you're going to get insulin. But below that, you have the 5' and you have the 3' ends, and then in between all of that, you have all these different combinations of the base pairs. Okay, you still have T and A and G and C, but because you have a different arrangement of these, and you have short and long chains that can be developed, then you're going to have a variety of different options. And his experimental methods, it's kind of like a brute force, where you just got to see all of the different segments that are produced, and then you can start identifying maybe which sequences are corresponding to a given protein. Okay, so these would be called like the codons, especially if they're in groups of 3. You have anticodons. You also have codons. We'll be talking about that in a minute. But if you want to get even more information about this, in case I'm not doing it justice, after all, I'm not a DNA specialist, but this DNA Learning Center has really great, sites and reference and schematics and cartoons and even animations that can provide additional, information. And again, I want you to just remember that RNA is used as a template for DNA, okay? And it does so because it's smaller, and that has incredible use. And so what we'll do, we will stop right now in this part of the lecture, in this part of the chapter. This is only going to be a 2-part chapter, and in part 2 tomorrow, we'll talk a little bit more about RNA.